carbon-carbon bond cleavage, irreversible in the cell, thermodynamically favored because you've dropped out CO2 as a product. And then we can go on from there. Alpha Kg complex like PDC we talked about, forming succinyl-CoA. Last thing we did last time was talk about succinyl-CoA synthetase, right, which is the enzyme that does a substrate level phosphorylation. This is succinyl-CoA here. Of course, because this is the alpha-ketoglutarate dehydrogenase complex, which is analogous to PDC, that means it's binding CoA as a cofactor. So here comes the CoA just like it does in PDC. CO2 is split off just like it is in PDC. And now your CoA is onto this substrate through a high-energy thioester bond, which can be used to make GTP. And CoA is split off just like it is in, it's lost in the PDC complex as part of the reaction. Here, the CoA stays on this intermediate of the TCA cycle and then comes off in the next step. Right? So in that respect, it is a little bit different from PDC. Okay. We then talked about how we get to succinate, and the carbons are racemized here in the sense that this is a symmetrical molecule, so that if we come in with a C14 label on these carbons of acetyl-CoA and we can trace it through to here, right? We note that the CO2s that come off are not the ones that came in on that round of the cycle. But then when we get to here, we note that this molecule is symmetrical, so you can bind either way on this succinate dehydrogenase, which effectively means that the carbon, a label with the carbon is now in all the carbons. Okay. And then the last steps are dehydrogenation, hydration, dehydrogenation. This three-step sequence is also found in fatty acid oxidation. Right, so that's worth bearing in mind. The first of these three steps is an FAD enzyme instead of an NAD enzyme. This is the one that's in the mitochondrial inner membrane because it's FAD. FAD does not diffuse. And so when FADH2 is formed from succinate, right? FADH2 is formed. FAD is going to go, these arrows are kind of cryptic, but the electron transfer reactions are succinate getting oxidized, FAD getting reduced. Now you have FADH2. Iron here, right, would be oxidized and get reduced because the electrons would come from FAD, right? So we go to iron 2, and then in turn that gets reoxidized, passes electrons to an oxidized form of this Q molecule, which is another redox cofactor that we'll talk about later. Okay, so this is sort of the beginnings of talking about the electron transport chain and how we make ATP. Making ATP is several steps downstream, so we're not talking about that yet. But we can note, here's the mitochondrial matrix, here's the intermembrane space of the mitochondrial. Here is NADH, and here this NADH diffused from enzymes in the matrix that produced it, that is to say, these TCA cycle enzymes, 3 of which are making NADH. Right? So that NADH comes over here and gets reoxidized to NAD while transferring electrons into a different set of carriers that goes into a different complex. You see this is labeled number 1, this is labeled number 2. So there's actually a different entry point in an electron transport chain for FAD, succinate dehydrogenase TCA cycle here, and all the other TCA cycle enzymes, NADH, and now it's coming through here. Okay? And that has an important bearing on why it is that NADH is worth 2.5 ATPs in that ATP arithmetic we looked at, whereas FADH2 is worth only 1.5. So we're going to go back over all this repeatedly. But just as an introduction to it, this is kind of part of the, this is the beginning of an explanation for why those numbers are different. Yeah.
This would be like going back one slide, but can you explain, or sorry, 2 slides. Can you explain one more time how the marker would be in all 4?
Yeah. So here it's in the 2, right?
Yeah.
And so you could say, well, if you just have one molecule of this, So I know that they're in these two, and these two look like these two, so I'm going to say for this one molecule, it's just in here, right? But what if you take this molecule and just spin it around, right? So that the substrate molecules are here. Now when I oxidize this further, right, it could be here or here. This is still symmetrical. But then when you go here, it's not symmetrical anymore, right? So once it's not symmetrical anymore, either the labeled carbons are here or the labeled carbons were here. But half the time they were here and half the time they were here. So by here, they're in all four, right?
But if you're just talking about one molecule, wouldn't it
Yeah,
still just
but we're not really talking about one molecule, so that's kind of the trick. We're talking about the progress through the cycle and how you would measure it.
Yeah.
So once we start talking about, oh, we're doing an experiment, and here's, C14 acetyl-CoA coming in, you're doing an experiment. So you've got a bulk sample, so you've got a gazillion molecules of this, right? Yeah.
So how would you measure it? How would you measure this radioactivity from this bulk sample that you've got that has been racemized?
Well, if you isolate the compound on TLC or HPLC, then you can do mass spec and get the isomers that way and see
Mass spec.
kind of where they are. Yeah. Any other questions about this? Brad, this kind of racemization of the carbon of the label into these four positions is a little bit not necessarily an easiest concept to get, but you have to think about it in terms of a billion molecules, and half a billion are going to have them here, and half a billion are going to have them here, and the same here. But by the time you get to here, the fact that this is asymmetric now because you've added water across the double bond, right? Essentially must mean, and this molecule bound on fumarase, either way, right? Then the label is going to be in all four of them, and we know that experimentally.
It's basically a mathematical average. Once you do the mass spec, it's not on all four of them, it's on half or half, but when you put it all together.
It's certainly diluted.
Yeah.
Right, so if you say that there's a certain specific activity, like a certain fraction of the carbon molecule, if I give you a label of acetyl-CoA, it's not all C14. It's like, 0.1% C14, 99.9%, C12, right? But that fraction will go down by half in these carbons here as compared to the fraction in those carbons there. Right? I know this is a little bit of a tricky concept, but the issue is just, and I put them on the quiz as well, but the issue is that the MCAT, for example, always tests on this kind of stuff. For whatever reason, asking about label on carbons is, it's a way of seeing that you guys can actually tracing through from, say glucose all the way through, so you see, what carbon is what and where the carbons go, which is a good way of kind of learning stuff, right? And so by saying, well, this one's labeled, it kind of emphasizes that you should be following it through, right? That's kind of the educational point. Any other questions? Okay. So the other reactions, talk about that. On this slide, it's simply an emphasis that when we hydrate fumarate to malate, these structures here are simply intended to indicate these are different stereoisomers, geometric isomers. So this enzyme is specific for this guy and will not touch malate. Okay, and if you're going in the reverse direction, notice that this is reversible. Malate is a substrate. Demalate, L-malate is not touched. Okay, so it's just to emphasize that enzymes are highly specific in that sense. They're highly stereospecific for chiral centers. They're highly specific for geometric cis versus trans. Right.
Is that showing an intermediate? I'm trying to follow that. Oh, it's showing the transition state.
Yeah, it's showing that there's. I don't know what it's showing. It's adding OH minus first. I don't like this. Forget about that.
Okay.
Yeah,
The point is just that it's specific for fumarate